Today we're going to talk about the basic concepts of energy transformation. That is, how we utilize the energy that we consume in our foods to do the work that we need it to do in our bodies. So we'll go into a lot more specifics when we go into the individual macronutrients later in the course. But for right now, we're just going to focus on how this process actually works on a cellular level. Okay, so our body requires energy to do just about anything. We need it to run, we need it to think, we need it to write, we need it to build complex molecules from simple precursors, such as building immune cells. We need it for active transport that we talked about in our last lecture, transport of genetic information. Pretty much anything you can think of requires energy that we need from fuel. So energy can be obtained from carbohydrates, lipids, proteins, and ethanol. And the primary source of storage energy in our bodies is adenosine triphosphate, or ATP. So here is the ATP molecule here, and we can see we have an adenine, it's adenine triphosphate. Adenine, we have a ribose, and we have 3 phosphates here. And the bonds of these phosphates are very high energy and store a high amount of energy that we can utilize for the work that we do. Okay, so ATP is primarily generated through cellular respiration. That is the oxidation of fuels to form carbon dioxide that we exhale and water. ATP is a major energy supplier in metabolism, and it's a really neat molecule because it serves as an energy reservoir because it stores all those energies in those phosphate bonds. And in that way, we can look at this little graphic here, and we can see we have ATP here, 3 high energy bonds, and these can be enzymatically lysed so that it would create ADP plus inorganic phosphate, plus release all of the energy that was stored in that bond of that terminal phosphate. So in that way, ATP is a primary intermediate for energy releasing and demanding chemical reactions in the cell because it allows us to utilize the energy when we need it. So energy is stored in ATP. Energy stored in ATP is released by the enzymatic hydrolysis and anhydride phosphate bonds. Now, when these phosphates are broken off, so the phosphate bond is broken, releasing the energy, that phosphate can then be transferred onto an acceptor molecule, and then that bond becomes high energy. So here's the ATP-ADP cycle, about as simple as it gets. So we have the ATP at the top, then we need to enzymatically lyse the terminal phosphate so that we can release the energy that we need for cellular work. And then what we have left is this ADP and this inorganic phosphate, but we no longer have the stored energy in that last phosphate bond. Then we consume energy from food, and it allows us to rebind this phosphate to the ADP to form ATP again, so that again we have this stored energy for when we need it. So ATP, next we're going to talk about the mechanisms by which ATP is formed. Now, counterintuitively, we start with phosphorylation, which is the transfer of a terminal ATP phosphate to a molecule. So we're kind of thinking, I thought we were going to create ATP from our fuels, but really we need this initial investment. Now, this step is called phosphorylation because we transfer a phosphate onto another molecule. And it's a 2-step process. So first we have the hydrolysis of ATP to ADP plus P, and then we have the addition of the phosphate onto a substrate. So one example that's very relevant in our field is when we have this phosphorylation with glucose. So enzymatic phosphorylation of glucose by ATP is the first reaction that glucose undergoes when it enters the cell. So we have the glucose, we have the ATP. ATP is broken up, so we just have the ADP, and the phosphate is added onto glucose to become glucose 6-phosphate, or G6P, which I'm sure you've seen when you've looked at the TCA cycle in the past. So then, glucose 6P has this high energy bond, and that can be stored as glycogen and oxidized for energy whenever we need it, such as when we do exercise or when we just haven't had enough energy input from fuel in a while. It can then release that energy that we need. So the lysing of ATP is a highly exothermic reaction in that it releases a lot of energy when that phosphate bond breaks. And that makes the endothermic reaction, that is, attaching the phosphate onto the glucose, energetically favorable. Because though it takes energy to bind that phosphate to the glucose, in this example, the reaction in total is exothermic and kind of goes downhill energetically, so it makes it energetically favorable. Okay, so as you would imagine, if we're breaking up all this ATP into ADP, then we've got to have a way to reform the ATP. We've got to be able to make it so that we have this energy storage when we need it. And this is highly controlled in the cell. So this homeostasis is highly controlled in the cell, I should say. So if we have not enough ATP, our bodies are going to try really, really hard. Our cells are going to try hard to make more. If we have plenty, then our bodies will kind of lay off a little bit and make less ATP. So ATP is formed through two mechanisms that we're going to talk about. So the first is substrate level phosphorylation, and it's exactly what it sounds like. It's the transfer of a phosphate group from a substrate that has this high energy phosphate bond onto ADP to make ATP again. So it's kind of the opposite reaction as the one we just discussed. So the phosphate is being put onto the ADP to make ATP. Now the one we talk about the most and is primary for ATP production is oxidative phosphorylation. And just like the title says, it requires oxygen and produces carbon dioxide. So part of oxidative phosphorylation is this electron transport chain, and it may also be called the respiratory chain. And this is why, because it requires the oxygen and creates carbon dioxide for us to exhale. And in this process, energy from the electron transport chain in the mitochondria are used to unite ADP with its inorganic phosphate to again make the stored energy that we can use. And we're going to go into a lot of detail about that. So before we move ahead, I want you to pause and see if you can describe the mechanisms involved with ATP formation. And I'm going to go ahead. First method, substrate level phosphorylation, I just want to touch on that because it is relevant, though it's not as common. So one important example of this is the anaerobic conversion of ATP. And as we can see, so it can't be oxidative phosphorylation because we don't have oxygen. It's anaerobic. And so we can only really utilize nutrients anaerobically with carbohydrate as the fuel. And in this process, we're able to provide energy for ATP production without electron transfer to oxygen that we are going to see that we really need in oxidative phosphorylation. And this is critical for cells with a decreased oxygen supply. So this could be if there was problems with respiration, if our arteries were clogged and we weren't getting as much oxygen to our cells, or if we were undergoing exercise. And we just can't consume enough energy that we need to, we just can't consume enough oxygen that we need for the energy. So in a situation like this, normally we would have glucose in our body go to pyruvate, which would go to the TCA cycle, an electron transport chain. But when we don't have enough oxygen, pyruvate can be converted to lactate, and that does not require oxygen. And that's why when we exercise, when we're really out of shape, we don't have enough oxygen, our bodies aren't used to the strain, we get this buildup of lactic acid. But when we can aerobically train ourselves to utilize our energy or our oxygen more efficiently, and therefore, when we continue to run, we don't have that buildup of lactic acid because our pyruvate is allowed to enter the TCA cycle. Okay, so now we're going to focus on our major topic, which is the TCA cycle and oxidative phosphorylation. Okay, so this process is always aerobic, and all fuels can be utilized for aerobic conversion of ATP, that is, conversion in the presence of oxygen. So let's take glucose because this is, the most prevalent nutrient that we consume. So first it undergoes glycolysis, then it enters the TCA cycle for oxidation. Glycolysis produces pyruvate, which enters the inner mitochondrial membrane and is converted to acetyl-CoA. Now, acetyl-CoA is very important because this is the common substrate that we need to enter the TCA cycle for ATP production. So here's a pretty rudimentary diagram. So here we have glucose, which again is converted to acetyl-CoA, and we can see that both fatty acids and amino acids can also be converted to acetyl-CoA. So this is our common substrate. Then it goes into the TCA cycle, the electron transport chain, and this is how our body produces ATP that we need for energy. So the TCA cycle produces over 2/3 of the energy from fuel oxidation. And again, acetyl-CoA is our common substrate that we need for this process. So here is our beautiful TCA cycle. So again, we see acetyl-CoA here, which we need in order to enter the TCA cycle. And then we have a series of steps that it goes through. So in each step, the substrate is being oxidized. Well, not in each step. Generally, the substrate is being oxidized as it goes through the cycle, and it's reducing an electron acceptor, which will then allow it to go to the electron transport chain to produce energy. So for the purposes of this class, I don't need you to necessarily memorize each step, but I need you to know what's happening here. So let's take a closer look at these steps. So we can see here's the substrates here, and here's the enzymes that are needed to make the process happen, make the reaction happen. And we can see here that one really common one is this dehydrogenase enzyme, okay? And then we'll come back to this figure, but at each of these dehydrogenase enzymes, we can see that we have a reduction of an electron acceptor, so the NAD, the NADP, or the FAD. So I'm going to call this part 1 of this lecture, and we will come back for part 2.